Welcome back. We're going to continue this lecture from where we left off last time and look inside the cancer cell to peer inside and see what really makes it a cancer cell. You'll recall there are two types of genes that get changed to make a cancer cell. The first type are oncogenes. They can be brought into a cancer cell in an active form, a turned-on form by a virus, or there are proto-oncogenes inside a cell. You remember 90% of cancers are not caused by viruses. These proto-oncogenes are normally dormant in a differentiated cell, and they can become activated by mutation. These are the genes that stimulate cell reproduction, the gas pedals. The other set of genes are tumor suppressor genes. They're the brakes for cell division. In the previous lecture, I described Knudsen's two-hit hypothesis. You remember, you need two mutations in these tumor suppressors to turn off the brakes and allow the cell to go forward. And these genes were inferred from studies of inherited cancers. You recall retinoblastoma. So these gene products normally block cell division, and when they are mutated in cancer, cell division can proceed. Now, like the case I made for viral oncogenes, you remember they led us to a description of proto-oncogenes in normal cells. We can ask the question, what about tumor suppressor genes? What do they do? That is, how do they act as brakes for cell division? With a description of these two types of genes, we will have a more complete view of cancer. I want to begin this discussion with the story of a woman I will call Ellen. This is a true case. Ellen is a 40-year-old woman who comes to her doctor asking about testing for what she calls the breast cancer gene. There's breast cancer in her family. Her sister, age 38, has just been diagnosed with breast cancer. An aunt died at age 52 with tumors in both breasts. Her grandmother died young as well at age 54 of breast cancer with multiple tumors. In Ellen's ethnic group, mutations in what I'll call for now the breast cancer gene are more common than they are in the overall population. In her group, it's about 2%. 1 person in 50 has mutations in this breast cancer gene, whereas in the overall population, it's much rarer, about 0.1%, which is quite a bit less. The physician interviewing Ellen and getting all this information says, in the family, there's early onset breast cancer. Well, most breast cancer happens after age 60, early onset. Multiple tumors in the family, multiple tumors in the breast, and in fact, in both breasts in one case. So he says, well, maybe we're dealing here with hereditary cancer. And maybe Ellen might be a person who, if she has this mutation, might be able to do something about it. We'll talk about that later on. The test that the physician orders is to get the entire DNA sequence, the nucleotide base sequence, A, G, T, T, C, C, of a gene called BRCA1. And to look for mutations not only in Ellen, but in other living members of her family, especially her sister who has breast cancer. Question: how did we get to this precise molecular test and this precise molecular description of a molecule and the possibility of a diagnosis? We got there thanks to some amazing genetics done initially by a professor, Marie-Claire King. Marie-Claire King was at that time a geneticist at the University of California, Berkeley. When she was a teenager at age 14, her friend died of cancer. And so this really prompted her to go into the field. She received training as a quantitative and molecular geneticist. And she decided to analyze DNA in families who had breast cancer and try to relate mutations in DNA to disease, that is, a mutation in DNA to breast cancer. So she looked at families that had inherited breast cancer. Now remember, our criteria for inherited breast cancer would be bilateral tumors, young age, tumors in multiple tumors in the breast, etc. And she tried to relate these DNA changes to the phenotype of breast cancer. Here is the argument. The argument is one of genetic linkage or linkage analysis. I mentioned that we are identical in 99.9%. It may be a little less, it may be a little more, but all humans are pretty much the same in their DNA. Well, wait a minute. If we're 99.9% the same and we have 3 billion nucleotides, that means there's a lot of differences between us at 3 billion. That's 1 in 1,000 to 3 billion. That's a lot of changes that are different. So what Marie Claire King tried to do was to do linkage analysis, to link a DNA change that was easily found, you can easily detect these things, to an inherited phenotype, that is, inherited breast cancer. And the tools to do this were ones that were available at the time. And the assumption was that if you can detect the linked gene, that is, the linked mutation, this one DNA change, it's probably somewhere near the breast cancer gene on the chromosome. So here's more or less what she could do. Here's a normal person who has at a certain spot a G in DNA. Here's a person carrying a mutation, which I will abbreviate C in DNA. Otherwise, in this particular region, the people are the same. Now we can look at a person inheriting 2 alleles, different forms of a gene, from the parents. One parent will give one allele, the other parent will give the other allele, and I'm just going to focus on the 3 letters around the G. So TGC, you can see here, and TCC in the mutant. So here's a father who is heterozygous. He has the normal allele, TGC, and he has an allele, TCC, as well. Mom is homozygous for the typical allele, TGC, TGC. One of the daughters is homozygous. She got the good allele from mom and the good allele from dad, so she's homozygous for the G at that point. But woman here, who might be Ellen in our example, inherited a C, TCC, from dad. And it so happens that this woman has breast cancer. You see the linkage? The linkage between a mutation that we can easily detect and breast cancer, whose gene we don't know about yet. Marie Claire King went around the San Francisco Bay Area for many years, painstakingly doing these DNA analyses on families with breast cancer. And by the mid-1990s, she had narrowed down to the location of the breast cancer gene to chromosome 17 of 23. You remember we have 23 chromosomes, and it was somewhere in chromosome 17 because a mutation, one of these mutations in DNA that's on chromosome 17, was present when people had breast cancer. She called the gene BRCA, you know what that's going to mean, breast cancer, and went looking for it. In the meantime, a team at a company called Myriad Genetics in Utah looked at her work and other people's work and looked for the gene. And the Utah team won. The man who headed the team is Mark Skolnick. His photograph is shown here. And there's a map showing the BRCA1 gene located at a certain spot on chromosome 17. I have to emphasize, this is very misleading because the size of the BRCA1 gene, is very much smaller, it takes up much less space on chromosome 17. If chromosome 17 was my height, the BRCA1 gene might be a pinprick, right? A small amount of the DNA. Be that as it may, we can kind of map it as to where the general area is and then isolate the gene. Then they looked at a lot of people and sequenced their DNA around on BRCA1, and they found mutations. And each little dot here indicates a person with breast cancer. There was huge optimism. Wow, it's fantastic. We have a breast cancer gene. And everyone wanted the retinoblastoma model to be true. Remember retinoblastoma? All people with the RB mutation have retinoblastoma, right? Mutation in the RB gene. Let's flip it around. People with retinoblastoma have the RB mutation. You see the difference? Was that true of BRCA1? In other words, people with BRCA1 had breast cancer. Was it true that all people with breast cancer have BRCA1 mutation? In other words, is BRCA1 the mutated gene in all breast cancer? That's what we were all hoping. No, it's not so. Sorry about that. At the most, it's 5% of all hereditary breast cancer. So people started looking for other genes, and they're still looking for them. They found the BRCA2, P53 mutations. There's a lot of different mutations involved with breast cancer. Is this surprising? Not really, because remember, cancer is caused by mutations in many genes. So you can figure that there might be a lot of different mutations involved. Second question we would like to know is, what about the majority of breast cancer, which is 90%, that is sporadic, not inherited? Is BRCA1 mutation mutated there? I'm sorry about that. It's not. It is in some, but it's extremely rare. But even so, we could test Ellen, because in her ethnic group, there's a fair number of people carrying a certain mutation in the BRCA1 gene. And she found that she did have the mutation, which indicated that she had a lifetime risk of 80% of developing breast cancer. Remember, she's much younger. She's 40 years old. And so she made a decision. And the decision was to prevent cancer. She had a surgeon remove both her breasts and ovaries. We'll talk about the cancer prevention by surgery later, but that's the decision that she made. The remainder of this lecture, I'm going to describe the strategies for identifying tumor suppressor genes. I've already described one of them. And then I'll describe what some of these tumor suppressor genes do. I'll describe how some of them repair DNA damage, and others controlling the cell division cycle, and finally others preventing DNA damage from becoming permanent. I'll give examples of all of these. So first, strategies to isolate tumor suppressor genes. And there were really 3 of them. Many tumor suppressor genes have been isolated at this point. The first one is DNA mutation linkage to the disease phenotype. Well, that's what I just described in the introduction with Marie-Claire King and Skolnick, isolating the gene by linkage to a single nucleotide difference between people. So I'm not going to deal with more of that. Second is chromosome analysis. These are bigger changes you can see under the microscope. You remember the opening story of the course? I described the basketball player Kareem Abdul-Jabbar, who had a translocation, a shuffling of the chromosomes between chromosome 22 and 9, 2 chromosomes that were shuffled. And this translocation was diagnostic of his disease. Well, sometimes DNA damage is not chromosome shuffling, but an actual missing part, a missing piece of a chromosome, and you can actually see this. So here is a really interesting series of people in terms of looking for a tumor suppressor gene. Here is the human genome. Here's all the chromosomes. And we are focused on one of them, chromosome 13. And now we ask the question, let's take a whole bunch of people and look at their chromosome 13s, and you'll find sometimes people who are missing a piece of chromosome 13. That's just what nature is. That you can get these chromosome deletions in DNA replication sometimes when DNA fails to replicate. So here is a person here, for example, who has missed all this part of chromosome 13. Does this person have retinoblastoma? Didn't. Here's another person who is missing a small piece of chromosome 13 down here. Did that person have retinoblastoma? No. Here's a person missing a piece of chromosome 13 here that's missing this part. And that person did have retinoblastoma. You see the reasoning? Nature, that is mutations, is doing an experiment for us. Nature is, through some accident, causing a deletion of the chromosome, and by mapping all of the deletions, we can figure out where retinoblastoma gene must be, where a tumor suppressor gene must be. Because if a person has a deletion of the tumor suppressor gene, they're going to have the disease retinoblastoma. And that's how they narrowed it down to a very small part of chromosome 13, and then were able to isolate it. And this has been done with other tumor suppressors. The third method of doing, so that's the second method of looking for tumor suppressors. The third is looking at isolating these genes, or we think is a tumor suppressor gene, and curing cancer by gene therapy. Well, I wouldn't say that. It's taking cancer cells in the laboratory dish, as is shown on the screen, and adding what we think is a tumor suppressor gene. So we take some breast cancer cells that have a mutation in BRCA1, and so they're going to be cancer cells. They're going to be behaving badly in terms of cancer on the laboratory dish. We've put them in a mouse. The mouse will get cancer. And then we take the normal gene, And we do a gene therapy. We add the normal gene to the cancer cells, and lo and behold, they become normal. And that was a way that people isolated more tumor suppressor genes. Overall, many tumor suppressor genes are now known. And I want to describe the functions of a bunch of them in more detail, but I'm going to give you an overview at this point. The first one on my list is BRCA1, and what that protein does is repair DNA damage. The tumors affected are breast and ovarian cancer. Now, I want to emphasize something here. All cells do not have the same genetic program for repairing DNA damage. Some cells use BRCA1, it's in the genome. Other cells, even though we have the gene for BRCA1, use other proteins for doing it. In the case of breast and ovarian tissue, they express BRCA1, and so they use it for repairing DNA damage. Tumor suppressor. It's going to be a tumor suppressor because the DNA will not be damaged and the cell won't become tumor if it's damaged at oncogenes. Cell division control. Well, we know about RB and retinoblastoma. The RB gene is also expressed, in addition to the retina of the eye, in bone and in lung. So RB acts in those tissues as a cell division suppressor. Checking for DNA damage. There is a gene which we will describe in detail in a few moments, called P53, and that's active in most cell types. Remember the cytoskeleton, this thing that allows cells to move and also give the cells shape. Remember there was a photograph I gave of the range cytoskeleton in cancer cells. There's a gene called APC that's involved with colon cancer that involves the control of the cytoskeleton, among other things. We'll talk about this later, next lecture. Regulation of differentiation. Remember, differentiated cells don't divide. So if you have a gene that controls differentiation in some ways in a tissue, and that gene is defective or mutated, then the cell will no longer be differentiated. It'll say, oh, I'm going to divide. So this is a case of a gene called PATCH, PTCH, in basal cell carcinoma of the skin. There are genes, tumor suppressor genes for cell adhesion. Here are the two cells sticking together. And this is the gene that codes for the protein that allows the cell to stick together might be mutated. And now the cells can separate, and they're going to be more cancerous. That's a CAD gene in stomach. You'll learn about another one in the next lecture. So let's look at a couple of these tumor suppressor genes and see what they do. And the first one I'm going to describe is a gene that repairs DNA damage. Now I'm talking about the three functions. Here is DNA damage due to radiation. It's severe damage to DNA repair. So if you look at this, there's DNA being damaged. You see the split of DNA. And the damage ultimately leads to cancer. A cancer cell is formed, you get growth, and you get a tumor. Okay? This is a great review of the previous lectures because it's really showing the centrality of mechanisms involving DNA. And the fact that you know that if DNA is damaged in cells, in genes controlling cell division, that these mutations can activate oncogenes or inhibit tumor suppressor genes. And these damage of DNA is caused by, to review, spontaneous mutations, happen because DNA is DNA, and things from the outside, carcinogens that are mutagens. So the question now is, what do BRCA1, and in addition there's another gene called BRCA2, what do they do? What does the BRCA1 do? A hint as to their role is to ask, when are these genes expressed? When do they make the BRCA1 protein? If we look at the cell cycle, when cells are dividing, we find that BRCA proteins are repairing DNA and active during the synthesis phase, when DNA is being made. So here is the cell cycle. You've seen this before. This is the restriction point, the point of no return. And after the restriction point, DNA is made during this time, this phase, and of course there's the cell dividing into two. During the synthesis phase, the BRCA1 protein, which before then has been hanging around the nucleus, or even outside the nucleus sometimes, just doing its thing, not being active at all, not repairing any DNA because it doesn't see any DNA damage, all of a sudden becomes changed and goes to bind DNA and zeroes in on DNA that's damaged. The change is a phosphate group, phosphorylate, adding phosphate from a protein kinase. You remember I've told you, protein kinases are extremely important in adding phosphate groups to protein. and they add phosphate groups to BRCA1 when there's DNA damage. When there's DNA damage, then the phosphorylated or phosphate-containing BRCA1 goes into the nucleus, binds other proteins, and they go and they repair DNA. So here's the repair going on. Here's the BRCA1. Here's a double-stranded break of DNA, for example. And the BRCA1 is adding phosphate groups, and that causes it to change shape, bind to a bunch of other proteins. Together, the gang goes into the nucleus and directs the repair to DNA damage, and the protein complex repairs DNA. Quite an impressive system. Now, this system works in breast and ovary cells. I have the gene for BRCA1 in every cell of the body, but it's only active in breast and ovary cells. It's not clear the reason why my skin cells do have different repair than the breast and ovary cells, but that's what it is. Okay? That's what it is in terms of cell differentiation. So in other tissues, other tumor suppressors work to do this same repair. So that's the first mechanism I wanted to talk about, which was DNA repair. Second mechanism I'd like to talk about is controlling the cell division cycle. Recall Knudsen, retinoblastoma protein, and we've seen retinoblastoma before. He predicted things about how it would act. It would act somewhere in the cell cycle to inhibit cell division. And as you know, he's right. I've described this in some detail before. We'll go in a little more depth today. And when the RB gene and protein were isolated, all of the predictions that Knudsen made turned out to be true. RB protein, you recall, acts at the restriction point, the point of no return. And what it does is, I said earlier on, RB acts at the promoter of genes involved in the expression, whose expression will make proteins that will stimulate cell division. So RB goes to the promoter, grabs DNA, right, and says stop, okay? That's what I told you earlier on in the course. Now we're going to get into a little more depth. It's a little indirect. You'll recall the control of gene expression that I described in an earlier lecture. Gene expression is controlled at this ignition switch, at the promoter. And there is a site for binding the promoter activator proteins. So here is an activator for transcription that's going to kickstart the transcription process. Remember last lecture I described at the promoter is a lot going on. There might be 15 proteins all piled up there telling the mechanism copy or don't copy the DNA, express or don't express. There is a protein which has a name, it's called E2F, but there's a protein that's an activator that activates cell division genes. And this is a protein that is turned off by RB binding. So in a typical normal cell, as a cell is hanging out, for example, during this phase of the cell cycle, it's not making DNA, it's not committed to do anything, it might be a differentiated cell, RB is bound to this E2F activator, and transcription shuts down of the cell division genes. If a cell is going to divide, RB is changed. Okay? RB is mutated or changed in some way, and RB could be phosphorylated, for example. The structure of RB changes, something happens to RB, or it's mutated, and E2F is now free to stimulate transcription. So you see the indirect switching on of DNA, where RB is no longer around to block it. RB is expressed in retina cells, in bone cells, and that's where you get the RB gene active. And when the RB gene is mutated, it doesn't bind E2F, and all of these things don't happen, and so E2F goes on. So here is something I showed you earlier, but in a slightly different way. So in cancer, if RB is mutated by a regular mutation of DNA, such that the RB protein can't bind to this E2F, this activator, the activator is free to stimulate cell division, it goes ahead and divides. Goes past that restriction point and goes ahead and divides. If RB is knocked out, RB could be knocked out by a virus. So a virus may knock out RB. That's how papillomavirus works. It binds to the RB, same thing's going to happen. In some tumors, a substance made by the tumor binds to RB and prevents it. Here's one of those substances. It's indirect. There's a gene called P16. RB gets inactivated naturally during a cell cycle, when our friend, maybe from the very early lecture on cell division, the cell cycle kinase, this enzyme, this protein that takes RB, which is blocking cell division, and puts phosphate groups on it, changes its structure so it doesn't block cell division and permits it to go ahead. So a growth factor may be stimulating the kinase. P16 is a tumor suppressor gene that makes a protein that binds to the kinase. Now if you can understand everything I'm going to say, you're doing really well, which is good. If P16 knocks out the kinase can't add phosphate groups to RB. If you can't add phosphate groups to RB, RB stays and the cell cycle is blocked. The P16 protein is expressed in a number of tissues such as pancreas, esophagus, and stomach. When P16 is mutated, imagine, the kinase is free to do its thing, modify RB, RB no longer binds, and of course, cell cycle goes ahead. So P16 is a tumor suppressor. When it's mutated, of course, you can get cancer. Now, why you say, why do you have all these control mechanisms? Well, because P16, for these tissues I mentioned, stops the cell cycle in differentiated cells. They stay differentiated, they stay specialized, and they don't divide. And they're going to have less DNA damage because of P16, because they're not dividing, and less cancer. So in a number of these cancers, in pancreatic cancer, stomach cancer, esophageal cancer, P16 is mutated, and that allows the cell cycle to proceed. Some tumor suppressor genes prevent DNA damage from being permanent. Let's go back to SV40, the monkey virus. And remember Dolbeck's model. SV40 infects normal rodent cells and can cause a tumor. It transforms them into a tumor. How's it do it? You remember there was a mutant SV40 that didn't transform, and there was a good SV40 that did. They compared the mutant and the normal, and they found the normal one had a gene, an extra gene. The gene is called T for transformation. It makes a protein called, which we call T. And so people ask the question, well, what does T do? The T protein is going to knock out tumor suppressors. Two people that did this were Arnold Levine at the Rockefeller University in New York and David Lane in Britain at the Imperial Cancer Research Fund. They went fishing. They transformed some cells into tumor cells, and they went fishing, so they had this protein present, right? They had the viral protein present, and they went fishing looking for a protein in host cells that the viral protein must knock out, a tumor suppressor it knocks out. And the protein they isolated was a protein called P53, and then they isolated the gene P53. Is this a gene that's involved with cancer? Yeah. If they breed some mice that have mutations in P53, the mice have big problems. So in this graphic, it shows that if a mouse has two alleles that are mutant for P53, it doesn't have this tumor suppressor, 75% get tumors within 6 months. You'll recall this family with inherited breast cancer, the Lee-Fraumeni family, and it showed that this family shows cancers in brain and breast and bone and leukemia and sarcoma. Why would that be? They all have the same mutation in P53, in the P53 gene, so they don't make a good P53 and it doesn't work. They get cancer, various cancers. What does P53 do? It's a Swiss Army knife. It does a bunch of different things, and to ensure that normally cancer doesn't happen. If there's DNA damage, a number of P53 gets activated, normally at a low level, and does three things: DNA repair, or apoptosis, or stops the cell cycles. And all of them involve preventing cancer. Get a sunburn. What happens? Skin peels off. Why? The sunburn damages DNA, sends a signal to activate P53. The P53 protein binds to promoters, stimulates the synthesis of proteins involved in programmed cell death. So what's going on when you get a peeling sunburn is the skin peels off because the cells have died in programmed cell death, and it's all due to P53. So P53 promotes apoptosis. P53 also arrests cells, less cell division. Same thing happens with DNA damage. And finally, in cell cycle arrest, in DNA repair, P53 looks around for DNA that's damaged, and it will help synthesize genes that are involved with DNA repair. How does P53 work? It binds to DNA. This is an amazing graphic. Here is P53, and here it is bound to DNA. And you can see there are little loops of P53 recognizing DNA. And now we're going to come back to something I showed you earlier on in the course. We talked about smoking gun and benzpyrene and lung cancer. You remember this graph? It showed you the P53 gene and where the mutations were where the cigarette smoke mutagen bound. Remember that? Right around here in P53 and right around in this different locations on P53. Well, look at the location where most of the mutations is, position 248. It's just where P53 touches DNA and does its thing to stimulate or modify transcription. Amazing specificity here. We've described how a mutagen, a single mutagen, that is benzpyrene in this case, binds to a specific region of a specific gene and changes the synthesis of a specific protein so it doesn't bind to DNA, which is P53. So you don't get transcription, you regulate transcription, and it's connected to the cell cycle and blocks the cell cycle. This is phenomenal detail about how cancer gets caused, from carcinogen to DNA to specific mutation to an inactive protein in tumor suppression. It's this kind of understanding that's going to lead to better therapies. So I've tried to describe in this lecture the strategies for identifying these tumor suppressor genes and how they work at the DNA level. The next lecture, we're going to put it all together, all this molecular detail, and see how these events play out as cancer develops. Thank you.